Good afternoon, everyone. It's really a great pleasure to be here. I'm going to tell you today about our product MB310 that we've developed for the treatment of osteocolitis. So we're going to change gear from cancer, but still within the realm of the microbiome. So Virginia gave a really brilliant introduction to my talk about the interactions between the host and the microbiome. And there have been a lot of work done, a lot of publications from different teams around the world studying or addressing this relationship between host and microbiome. But what we know is really the tip of the iceberg. There is a lot of things under the water still need to be understood and characterized in this interaction. So, as Virginia mentioned, the interaction takes place through metabolites, through proteins or components of the microbial wall. But even within the metabolites, short-chain fatty acids, for example, the effects of these metabolites can vary depending on the physiological condition of the host, whether it's healthy or disease. They can be helpful in one scenario, not really helpful in another scenario. They can have certain effect in one case, but completely opposite effect. And we have a lot of stories around that. So that's just one case of a group of metabolites. So another factor is the microbial community. These bacteria, they talk to each other and they talk to the environment around them. So all of that has an impact on their functionality. Therefore, in our strategy to develop our products, we didn't really depend on the characteristics or functions of the microbes in the microbiome, but we adopted a clinic-first approach. So how does that work? We characterize the microbiome composition of big patient populations using our very unique platform. I'll talk about that in a bit. And from that, we identify specific microbiome signatures that are correlated with a certain clinical outcome, like a certain type of therapy, immune check inhibitor, for example, in cancer, or fecal microbiota transplantation, ulcerative colitis. I'll talk about that in a bit. So we address a certain clinical question within a group of patients, and we correlate that microbiome signature to that clinical question. And from that microbiome signature, we can develop biomarkers to stratify patient populations that can receive a certain type of therapy or develop a live biotherapy product. In order to do that, it is very important to identify the microbial species you have within the microbiota to very precisely and to a very high resolution. Usually with a microbiota, let's say, have an example, 100 species. How do you identify the bacterial species and their relative abundance? Usually with metagenome analysis, where you have break down the DNA metagenome to hundreds of millions of short sequences, DNA sequences, and then you map them against a reference genome database. And here, the reference genome database is very, very important. The more comprehensive and deeper that database, the more species you are capable to identify, the more precise the microbiome characterization is. So it really pinpoints the importance of the reference genome database in use. And that's what sets Microbiotica apart from other companies. In the field, we have our own reference genome database. It's actually physical, comprises physical, not in silico assemblies like other companies. And it is essential for high precision identification of bacterial species. And we keep on expanding this database. We keep on adding to it to remain on top of it. In addition to the reference genome database, we've developed in-house our own bioinformatics capabilities that would allow us to identify these signatures. We're also very good with microbiology skills and with isolating bacteria from different patients. We have personalized bacterial banks from each patient. That led us to a physical collection, bacterial collection of over 100,000 different bacterial species and strains. Each one of them can be a potential therapy. So for our pipeline at the moment, we have 2 clinical programs. Really happy about that. We started both trials late last year. One in melanoma immunotherapy, so checkpoint inhibitor resistant melanoma with MD007. The second one is with ulcerative colitis using MD310. We also have other discovery programs, including the barrier repair program, other indications. On top of that, we're also conducting a MITRE observational study in cancer patients in collaboration with Cancer Research UK. But we're going to mainly focus on MD310 and ulcerative colitis in my talk today. So just to give you a bit of a background on ulcerative colitis, as you may know, microbiome plays a very important role in the pathology of UC. In inflammatory bowel diseases, it's been very well characterized that you have a dysbiotic microbiome, and the composition of the microbiome has a very important role in the severity, the clinical severity of the disease and response to different types of therapies. Fecal microbiota transplantation from UC patients into mice leads to actually activation or induction of inflammatory type of T cells, and thereby predisposing these mice to development of colitis. What's really, really interesting in the past few years have been several FMT studies in UC patients. So fecal microbiota transplantation from healthy individuals to UC patients that shown quite some clinical efficacy in inducing remission in these patients. From these studies here, I'm going to focus on one that had been conducted by Sam Costello at the University of Adelaide, down under. He did a randomized placebo-controlled trial. In the active arm, he gave mild to moderate mild UC patients FMT from healthy individuals, either by colonoscopy or by enema, and in the placebo arm, the patients received autologous FMT as a control. And he looked at steroid-free remission in these patients at 8 weeks and also follow-up at 12 months. And this trial resulted in 1/3 of the patients in the active arm showing remission at 8 weeks. Half of them had persistent remission at 12 months follow-up. So it's really interesting. So we started a collaboration with Sam, got the stool samples from patients that went through the trial in the active arm. And as I said, in the active arm, you had responders and non-responders. We looked at the microbiome composition in both patient groups, and we did a profiling and consortium selection using our own platform, which led to MD310 consortium. I'll give you a bit of a short look or summary look on how we've done the kinetics analysis. This is a very simplified view. A lot of work had been put in this, but the first step was identifying the engrafting species with the clinical response. And then from that point, we selected multiple consortia with improved association to response. And that led to building a consensus consortium representing all taxonomic groups. And from that, we finalized the consortium and defined it as species. So with that, we developed MD310, which is a consortium of 8 different species, covering the major health-associated bacterial phyla, so Actinobacteria, Bacteroidetes, and Firmicutes. They are all novel species, only identifiable using our own platform. They're fully characterized bacterial species, done process development and preclinical development internally and also using external capabilities. And we have a lyophilized GMP product for oral delivery. Just to give you a bit of an idea on the preclinical development we've done for MB310, we've done a thorough safety profiling of these MB310 strains. First of all, they're very, very pure. They're all derived from healthy individuals, so no association with disease whatsoever. They have a really good antibiotic resistance profile. So at any given point, all of these 8 strains respond to 4 different antibiotics. So if there is a concern during the treatment, we can kill these bacteria using any of these 4 antibiotics. And also, no antibiotic resistance was identified on mobile elements, meaning antibiotic resistance cannot be transferred to the local or resident microbiome within treated patients. And they have also low pathogenic potential, so no harmful virulence factors or toxins. From a developability point of view, also no bacterial phages were detected in these bacteria. This is really important from a production point of view. So with all of these specifications, we deem these strains safe as per guidelines. Comes to manufacturing, this is a very oversimplified slide for manufacturing. It was a very exhausting process. Our guys in process development did a really great job. So we've done fermentation of these bacteria, so growing them to a larger scale under anaerobic conditions. Then we went to concentrate them and removing the buffer, then lyophilizing them. So every individual strain was, for these 3 processes, it was done separately, and then mixing them all together, these lyophilized material, and then filling them up in enteric capsules, oxygen-free enteric capsules, ready for delivery for the patients. And that's what happened late last year when we launched our phase 1b clinical trial, Composer 1. It's a randomized placebo-controlled double-blind trial in mild to moderate UC patients. So that's exactly the same patient population we started with in developing MB310. And the major or the primary outcome, first assessing the safety and tolerability of MB310. Secondary endpoint is looking at the engraftment of MB310 strains in the gut of the patients, and also looking at early signs of clinical phase. So as you can see here, we have 2 arms, screening, randomization. Both arms, patients in both arms receive vancomycin preconditioning, followed by a washout period. In the active arm, patients will receive MB310 daily plus standard of care. In the placebo arm, they're going to receive placebo daily over a period of 12 weeks, followed by a 12-week follow-up period. So the trial is ongoing, still recruiting. We have randomized patients that receiving the treatment or the placebo. So how does MB310 work, or how do we think MB310 is working? First, I'm going to remind you a bit of the cellular responses in ulcerative colitis and ulcerative colitis pathology. In normal physiological conditions, you have a healthy microbiome. This is a very elusive term. You have healthy mucosal and epithelial barrier, and also the immunological scene, both innate and adaptive, is dominated by tolerogenic responses. However, in ulcerative colitis, as I mentioned earlier, you have a dysbiotic microbiota, and you start having damage in the epithelial barrier. This damage will trigger some alarm signals in the epithelial barrier, leading to the release of inflammatory cytokines and chemokines, attracting inflammatory immune cells. But also, this leakiness leads to direct interaction between the microbiota, or these leaking bacteria or their metabolites, with the innate immune cells, the macrophages, and the dendritic cells, which start producing inflammatory cytokines like TNF, and they also start interacting with the T cells here, leading to the priming of inflammatory T cells like Th1 and Th17. So it's an inflammatory dominated site. So we started looking at the effect of MB310 at each of these cellular compartments. And we started with looking at the effect of MB310 on epithelial barrier. For that, we used a very standardized model with microtube cells. It's a cell line, a colon cancer cell line. It's a epithelial cell line. And we usually measure the electric resistance of a monolayer of these cells, of these cultured cells. If the electric resistance drops, that means you have more holes in that monolayer, meaning the barrier is compromised. If it goes up, that improves, or meaning improvements in the epithelial barrier. And we took controls, so we had Salmonella as a negative control, known to compromise the barrier. We took Faecalibacterium F. prau as a positive control, known to support the barrier function. And testing the different MB310 strains, we found that 3 of our strains are actually improving the barrier function by increased electric resistance. One of them more or less to the same level as F. prau. But what was really more interesting is that when we treated the caco cell monolayer with LPS, which mimics the inflammatory conditions or the conditions under which the barrier is compromised in ulcerative colitis, 3 of our strains mediated the healing and the repair of the epithelial barrier. This is very, very exciting. We started investigating the mechanism of action. Can tell you briefly that we've identified 1 novel protein and 2 metabolites involved in this process. I hope maybe next time I'm invited here or securing another invitation to be able to discuss that in more detail. So that's one thing done, barrier function. Then we moved on to look at the effect on innate immune cells, namely dendritic cells and macrophages. We used human monocyte-derived DCs and macrophages, so to isolate monocytes from blood, from human blood, and with certain growth factors, you differentiate into either dendritic cells or macrophages. And we looked at the production of 2 cytokines, TNF as an inflammatory cytokine, and IL-10 as a more tolerogenic cytokine. And you can see here that some of our strains actually promote the production of IL-10 to a much larger extent than TNF in both dendritic cells and macrophages, meaning that these strains are skewing dendritic cells and macrophages towards a more anti-inflammatory profile and program. So here we also have a positive effect on innate immune cells. In both setups here with the innate immune cells and also the epithelial barrier, we used the whole bacteria. We tried to maintain a live interaction between the cells and the bacteria in interrelated condition as much as we can. But when we move to T cells, we tend to looking at the metabolite, bacterial metabolites, rather than the whole bacterial cells. And here we had 2 setups. The first, looking at the effect of MB310 metabolites on dendritic cells, again, monocyte-derived. Then washing these dendritic cells and co-culturing them with allogenic CD4 T cells. And then looking at the profile of these CD4 T cells, whether they express any markers for regulatory T cells, which are responsible for maintaining tolerance in the gut. And indeed, 3 of our species produced metabolites that led to the induction of FOXP3 positive Treg cells, and 2 of them led to the induction of IL-10 producing regulatory T cells. So we have 2 different types of regulatory T cells being induced by MB310. Furthermore, when we look at the function of these regulatory T cells, so in addition to phenotype, the hallmark, the functional hallmark of regulatory T cells is their capacity to suppress bystander T cells. And when we looked at in a Treg suppression assay, 2 of our species, through their metabolites, led to the induction of suppressive regulatory T cells. So it's not only the phenotype, but we actually have fully suppressive regulatory T cells. So meaning that when we induce Treg cells in the gut, they are capable of suppressing the effector T cells here, the Th1 and the Th17. Another setup we looked at is looking at the effect of MB310 metabolites directly on T cells. So the metabolites here comes in direct interaction with the T cells while being primed by the dendritic cells. And with this setup, we also found three bacterial species that induced OXP3 positive Treg cells. From them, one did induce fully suppressive regulatory T cells. So we have different phenotypes of Tregs being induced and also through different mechanisms, either indirectly through dendritic cells or direct effect of these metabolites on the. So with this, we believe that MB310 will be beneficial by tackling key aspects of UC pathology. So by restoring the compromised epithelial barrier, by skewing innate immune cells, namely dendritic cells and macrophages, towards a tolerogenic profile, producing more IL-10 than TNF, and also the active induction of regulatory, suppressive regulatory T cells, different phenotypes that are capable of suppressing the bystander inflammatory. So to sum it up, I told you about our strategy in Microbiotica in developing live biotherapeutic products using a clinic-first approach. That's how we developed MB310, a consortium of 8 engrafting bacteria that act by tackling different pathologies or key aspects of UC pathology. And most importantly, we started our first in-human phase 1b trial last year, Composer 1, hoping to conclude this trial, have some data towards the end of it. Obviously, this has not been done by myself. Involved the efforts of a lot of people, both former and current team members within Microbiotica, and different teams from microbiology to bioinformatics. And my team, translational biology, did a lot of work in understanding the interaction with the host cells. And also big thank you to our collaborator, Sam Costello at Adelaide. And definitely a big thank you to all patients that participated in former studies and also current studies. Without them, we wouldn't be able to do any of that. Thank you very much for your attention.